Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic sexual reproduction in the flowering plants. Okay, so this lecture is the continuation of the previous lecture. So in the previous lecture we studied about the pre-fertilization, fertilization and the post-fertilization events. Okay, so pre-fertilization is the formation of the gametes, and during the fertilization what happens? The male and the female gamete, they fuse with each other. So we studied about the double fertilization, that is the characteristic features of the angiosperms. Okay, and then we studied about the post-fertilization events. So what we studied in the post-fertilization events, or what happens after the fertilization? Endosperm development, and then there is the embryo development, and then the ovules, they form the seeds, and the ovary, they get transformed into the fruit. Okay, and one of the characteristic features of the angiosperms, which are also known as the flowering plant, is that the seeds are enclosed within the fruit. So in this lecture, let's start with this lecture. So in this lecture we'll be talking about the Apomixis and the Polyembryony. So what is Apomixis? What is Polyembryony? Okay, and then we'll also study about the hybrid. Why we produce hybrid and what is the procedure of producing the hybrid, we will discuss that in brief. Now let's start with the lecture. So although the seeds in general, they are the products of fertilization. So seeds and the fruits, they are the products of the, you can say, sexual reproduction, because fertilization takes place during the sexual reproduction. And after the fertilization, what happens? So you can say after fertilization, or post fertilization, what happens? Ovule develop into the seeds, and ovary, they develop into the fruits. Okay, clear? Now all the seeds in general are the products of the fertilization. A new flowering plants, such as some species of the Asteraceae and the grasses, have evolved a special mechanism to produce seed without the fertilization, and this is known as apomixis. So what is apomixis? The production of the seeds without the fertilization, means without the fusion of the male and the female gametes, the seeds are being produced. Okay, generally the seeds are produced from the mature ovule. Mature ovule develop into the seeds, but in this case what is happening? Without fertilization, seeds are being produced. So this is a special kind of mechanism. Okay, so it is a kind of asexual reproduction. Okay, apomixis can be said as a type of asexual reproduction. So some flowering plants, such as, which belong to the Asteraceae, as well as the grasses. So grasses belong to the monocots. Okay, in these plants, apomixis is seen, means here the seeds are forming without the fertilization. Now seed production by apomixis involves the formation of seeds without the process of fertilization. This we have already discussed. So instead of a typical sexual reproduction involving the fusion of the male and the female gamete, apomixis generates seed that are genetically identical to the parent. Okay, so what you have to keep in mind, we are talking about the apomixis. So what is happening during the apomixis? Okay, seeds are forming. Seeds are formed without fertilization. Then you can say it is a kind of asexual reproduction. Why you can say it is a called type of asexual reproduction? Because there is no fusion of the gametes. There is no fusion of the gametes happening here, means there is no fertilization. Fertilization takes place during the sexual reproduction. Clear? That's why you can say apomixis is a kind of asexual reproduction. And here what is happening? Seeds are being generated that are genetically identical to the parent. This also is an important point. Here the seeds which are produced, they are genetically identical to the parent. Okay? Why they are genetically identical to the parent? Again, because there is no fusion of the gametes. There is no fusion of the gametes, so the seeds are genetically identical to the parents. During gamete formation, you know what happens? Meiosis and the recombination happens during the gamete formation. So during gamete formation, which cell division is involved? Meiosis, and meiosis involved is crossing over and the recombination, and this leads to the formation of new genome. Okay, you can say formation of new genetic makeup. Okay, and after the fusion also, what happens? Both the genetic makeup of the male as well as the female, they are mixed together. So during apomixis, there is no fusion of the gametes. So these seeds are genetically identical to the parents. There is no mixing of the, you can say, the genome. Clear? Now, what is food production without fertilization called? This you should know. It is known as the parthenocarpy. The production of the food without fertilization, this is known as parthenocarpy. Okay, and the fruits are known as parthenocarpic fruit. Fruits are known as parthenocarpic fruits. Okay, and these fruits are seedless. The seeds are not viable even if they are produced. So these fruits are seedless. So production of fruit without the fertilization, this is known as parthenocarpy. Now, so thus we can say apomixis is a form of sexual reproduction. So in sexual reproduction also what is happening? Okay, the progeny which is produced, it is similar to the parent. So in asexual reproduction, what happens? The progeny is genetically similar to the parent. Genetically similar to parent. Okay, clear? So apomixis, in apomixis also same thing is happening. The seed which are produced, they are genetically similar to the parent. Now there are several ways of development of the apomictic seeds. Okay, these seeds can be developed in several ways. Let's have a look at them. Now apomixis, okay, generate seed that are genetically identical or highly similar to the parent. That's why we call apomixis a kind of asexual reproduction. And in asexual reproduction, one more characteristic of asexual reproduction is it is uniparental. Okay, there is no fusion of gametes. Okay, so all these characteristics are seen in this apomixis. Now this reproductive strategy bypasses the formation of the embryo through meiosis and fertilization, and instead involves the development of the embryo from the maternal tissues within the ovule. Clear? So this kind of strategy bypasses the formation of the embryo through meiosis. Okay, so what happens? There is no fertilization, there is no fusion of gametes. Okay, and gametes are formed through meiosis. Okay, meiosis. So let's talk about the gamete formation, gametogenesis. Okay. Gametogenesis is the gamete formation, and gamete formation, it involves meiosis. Okay, clear? So no gametes are formed. There is no, so during the, you can say, asexual reproduction. So apomixis is a type of asexual reproduction. No gametes are produced. Okay, clear? So there is no fertilization. Okay, so the progeny produced is similar to the parent. Clear? So this reproductive strategy bypasses the formation of embryo through the meiosis. So usually what happens, how embryo is formed? There is one male gamete, and then you have the female gamete. Both these gamete, they fuses with each other to form the zygote, which is diploid structure. Both these gametes are haploid, and then the embryo is formed. Clear? Which will also be diploid. So this happens during the sexual reproduction. Or the normal embryos, these are formed like this. But during the asexual reproduction or during apomixis, what is happening? The embryo is formed, okay, without forming these gametes. Okay, so no meiosis is involved here, and no fertilization is involved during the apomixis. Clear? Now, let's talk about apomixis. Okay, how apomixis takes place. First is the initiation. So apomixis can be initiated through various mechanisms, including the development of unreduced egg cell. Okay, so egg cells, they are usually haploid. But sometimes what happens, unreduced egg cells are formed. So these unreduced egg cells are diploid. Okay, now the somatic cells or other reproductive structures, apomixis can be initiated. Let's read it again. Apomixis can be initiated through various mechanisms, including the development of unreduced egg cell, somatic cell, and other reproductive structures within the ovule. Now let's talk about this unreduced egg cell, which will be diploid. Okay. Now one common pathway of apomixis involves the production of the unreduced diploid egg cell within the ovule. Okay, within the ovule, what is present? Within the ovule, the female gametophyte known as the embryo sac is present, and within the embryo sac, this egg is present, which is usually haploid. Okay, but sometimes diploid egg cell is formed, which is known as unreduced egg cell. This can be produced due to the problem in the meiosis. Okay, so if there is a problem in the meiosis and if there is no separation of the chromosome, then the ploidy level of the cell will increase. Okay, the chromosome will not separate, then one of the cell, it will contain all the chromosomes. Clear? Now, one common pathway of apomixis involves the production of this unreduced egg cell within the ovule. Instead of undergoing meiosis to produce the haploid gamete, certain cell within the ovule may bypass meiosis and directly develop into diploid egg cell. Okay, so there is no meiosis. Okay, or you can say there is some problem is meiosis, or there is some disruptive meiosis. Meiosis usually leads to the reduction of the chromosome. So they can bypass meiosis, or what can happen? The chromosomes are not separating during the meiosis. Clear? That leads to the formation of a diploid egg cell. Now let's talk about the embryo development next. Now without fertilization, the unreduced egg cell within the ovule can develop into an embryo through the process such as the mitotic cell division. Now unreduced egg cell has been produced, okay, which will be diploid. then it will undergo mitotic divisions to form the embryo. Okay. Now this embryo development occurs within the ovule and leads to the formation of a seed containing a genetically identical or highly similar embryo to the maternal parent. Clear? So directly from the unreduced egg cell, the embryo is formed. So embryo will be again diploid. Now next is the endosperm development. Now in some case of apomixis, the development of the endosperm, the tissue that provides nutrients to the developing embryo, may also occur without the fertilization. Now this condition contributes to the formation of a fully developed seed. Now next is the seed formation. The seed matures within the ovule and becomes surrounded by seed coat tissues. Now once fully developed, the seed may be released from the ovary as a viable seed ready for the germination as well as the plant growth. Okay, this is clear. We were talking about the apomixis and how the embryo develops without the fertilization. Okay, so one of the most common reason is the what? Formation of the, we're talking about the apomixis. Okay. One is the formation of the unreduced, okay, egg cell. Unreduced egg cell which is diploid. Okay. Now this egg cell, these divide by mitotic divisions. And then it will form the embryo, which will again be diploid. Now let's move on. Apomixis, again, apomixis can occur naturally also in certain plant species, or it can be induced artificially also through techniques such as tissue culture or genetic modification. Now it is an important reproductive strategy in the agriculture as it allows for the clonal propagation of desirable plant varieties without the need for pollination or the involvement of the male gametes. So by apomixis, or what is the importance of apomixis? Here, the desired plant can be propagated. Okay, clonal propagation can be done of the desired plant without the pollination, means without the transfer of the pollens. Okay, what is pollination? Transfer of the pollens from the anther to the stigma. So there is no need of pollination here, and there is no need of male gametes. Okay, because the pollens, they form the male gamete. Pollen itself is known as the male gametophyte. So this is the, you can say, importance of the apomixis. Apomixis can be induced artificially also, and it can be used in the clonal propagation of the desired varieties. However, apomixis can also limit the genetic diversity within the population and may restrict the ability of the plant to adapt to the changing environment. So during apomixis, what is happening? Okay, the parents are, the offerings or the seeds are, they are genetically similar to the parent. Okay, so there is no recombination having, no crossing over is happening, and that may limit the genetic diversity. Okay, because if two parents are involved, okay, then if two gametes are involved of the two different parents, then after the fusion of the gametes, they will receive the genes from both of the parent. Okay, but here in apomixis, there is no fusion of the gametes, and that's why it can limit the genetic diversity, and this can affect the survival of the plant. So it can, okay, so it can restrict the plant ability to adapt to the changing environment because the genetic diversity is less. The type of the genes are very less. Okay, so more the diversity, okay, more the genetic diversity, you can say, okay, better the plant will be able to adapt to the changing environmental conditions. Okay, clear? Now let's move on. Now, Occurrence of more, now let's talk about the polyembryony now. Occurrence of more than one embryo in a seed, this is known as the polyembryony. This is very simple. Poly means many. So occurrence of more than one embryo in a seed is known as polyembryony. What are the causes of the polyembryony? Let's talk about these causes of the polyembryony include nucellar and the integumentary embryony. What is this? Embryo developed from the cell in the nucellus or the integument tissues surrounding the ovule. Okay, so these nucellar cells, these are diploid. So here what happens? Embryo can develop from the nucellus or the integument. Okay, this is known as, okay, clear? So this is the reason of the multiple embryos. Next is the adventitious embryony. Embryos form from the somatic cells of the ovule or the surrounding tissues. Okay, this is known as adventive embryony. Okay, formation of embryo from nucellus and integument, this is also known as adventive embryony. Okay, so these nucellus, integument, these are all somatic cells. Now hybridization, interspecific hybridization can lead to polyembryony. Okay, interspecific means between the species. Okay, if you make a hybrid between the species, then it can also lead to polyembryony. Next is genetic mutation. The mutation in the genes involved in the embryo development can disrupt the normal process. Clear? And next is the environmental stress. Now stress conditions such as the temperature, extreme temperature or nutrient deficiency can trigger the formation of the multiple embryos. Okay, so environmental stress can also lead to the formation of many embryos, which is known as polyembryony. Now, what would be the genetic nature of apomictic embryos? Okay, now can they be called as clones? Now what is the answer? Let's discuss about the answer. What is the nature of the apomictic embryo? So now you know these embryos developed from the unreduced egg cell. Okay, without the fusion of the gametes. So apomictic embryos typically have a genetic nature that is identical or highly similar to the maternal parent. Now this is because apomixis by definition involves the production of the seed without the fertilization, bypassing the genetic recombination that occurs during the sexual reproduction. Clear? Genetic nature means they are very similar to the parent, especially the maternal parents, or you can just say they are genetically similar to the parent. The genetic makeup is generally similar to the parent. Why? Because they lack the formation of the gametes and the fusion of the gametes. So formation of the gametes is because of the meiosis, and meiosis involved the crossing over and the recombination. So here I've written, this is because the apomixis by definition involves the production of the seeds without the fertilization. Clear? Now next, hybrid varieties. Let's talk about the hybrid. Hybrid varieties of several of our food and vegetable crops are being extensively cultivated these days. Okay, why we are using hybrid varieties? Because these hybrid varieties, they have increased productivity. Okay, because of the lack of the food, now we have to focus on growing the hybrid varieties. Also, these hybrid varieties have better vigor. Okay, they have better vigor also, they have high productivity also, and they are tolerant to a lot of stress also, both abiotic as well as the biotic stresses. Okay, so the cultivation of hybrid has tremendous have a potential to increase the productivity. Okay, the yield will be more, or you can say the yield will be more. Okay, hybrid consists of the hybrid vigor. Okay, means its productivity will be more, yield will be more. Okay, and it can tolerate a lot of stress as well. Now let's talk about the hybrid seed production in just briefly we'll discuss. First is the parental line selection. Now two parent lines with complementary traits are chosen. based on their desired characteristics such as high yield, disease resistance, or the quality traits. Here, first is the parent line selection. So, two parent lines with the complementary traits are chosen, okay, based on their desired characteristic. One of these characteristic is high yield. We want high yield. So, this characteristic is chosen. Now, second is the isolation. The parental lines are isolated to prevent the unwanted cross-pollination from other plants of the same species. Clear? So, these parental lines that you have selected, okay, these are protected from the cross-pollination from the unwanted pollens. Clear? So, one of the technique is you can bag it. Okay, bagging can be done. Okay, and if the flower is bisexual, then emasculation can be done. What is emasculation? Removal of the anthers, and then what you do? You pollinate the plant with the pollen, okay, that you have selected, that have all the desired character. Okay, so that pollen will be from that male plant, okay, from which you want to choose the characteristic. Okay, or you can say pollen from the desired plant can be used. Okay, and then again bagging is done to prevent the unwanted cross-pollination. Okay, so emasculation is done. So male reproductive organ, which are known as the stamens. Stamens consist of filament and the anther. So male reproductive organ of one parent are removed to prevent the self-pollination and ensure the controlled pollination. Clear? So first method is the first step is the choosing the parental cell lines. Next is the isolation. We isolate it, or we can also bagging can also be done of the flowers. Okay. Now next is the emasculation, means removal of the anther. Okay, removal of the anthers is known as the emasculation, and this will prevent the self-pollination. Okay, this is usually done in the bisexual flowers. Emasculation is done, but in unisexual flower there is no need of emasculation. Okay, we can just pollinate that with the plant. Okay, we can just pollinate them with the pollens of the desired plant, and then we can bag it. Okay, so in bisexual flowers, emasculation can be done. Clear? Emasculation, it is the removal of flower and is the method to prevent the self-pollination. What is self-pollination? The transfer of the pollen from the anther to the stigma of the same flower. Okay, self-pollination, or you can say autogamy. Autogamy it is self-pollination. What is self-pollination? Transfer of the pollens from the anther to the stigma. Okay, transfer of the pollen from the anther to the stigma of the same flower. So this happens in the bisexual flowers. Okay, now next, hybrid seed production. Next is the pollination. The pollen from the selected male parent is transferred to the stigma of the emasculate female parent to facilitate the controlled fertilization. Okay, so now the pollens from the desired plant are dusted, okay, over the stigma of this emasculated female. Now next is the seed development. Now the fertilized flower develop into the seeds containing the genetic material from both the parents, resulting in the hybrid seed with the combination of the desired character. Okay, now the seed that will be produced, okay, these are known as the hybrid seeds. Clear? So the method is clear. So what they have done, they have selected some lines. Then what they have done, if the flowers are bisexual, then what you can do the emasculation. You can remove the anthers. Okay, and then you can what you can do, you can dust this flower, okay, which is emasculated with the pollens of the desired male parent, and then you again bag it to prevent the unwanted cross pollination. Clear? Now then you can do, if now when you dust the pollens from the desired male plant, then fertilization will take place and there will be a recombination. Okay, there will be the fusion of the male and the female gametes. So male gamete is produced by the pollen and female gamete is produced in the embryo sac. So both of these will fuse and there will be the, okay, and this fusion will result in the formation of the hybrid seed, which has the combination of the desired characteristic. Clear? So we can write, first is the pure line selection. Okay, next is the, you can say, isolation of the flower, or you can do bagging also. So next is, this is to prevent the cross pollination. Okay, to prevent contamination from unwanted pollen. So you can say contamination or pollination. Next is the emasculation. Then we have the pollination. Pollination from the pollen of the desired, pollen is acquired from the desired parent. Okay, and then after it, fertilization will take place, which will lead to the formation of the hybrid. Okay, clear? Now, one of the problems of hybrid is that hybrid seed have to be produced every year. Okay, this is the problem in the hybrid seed production. Now, if the seed collected from the hybrids are sown, the plants in the progeny will segregate and do not maintain the hybrid characteristic. Okay, so in next generation, what happens? There will be segregation of the characteristic. So the desired characteristic may be lost. Alright, so there is a need to produce this hybrid seed again and again, because whenever they go to the next progeny, there will be segregation. You must have studied about the law of segregation of the mental. Okay, so there will be segregation of the characteristic and the desired character won't be there. Now, next is the production of the hybrid seed is very costly, and hence the cost of hybrid seed become too expensive for the farmer. So these hybrid seeds are very costly. Now, if these hybrids are made into apomix, there is no segregation of the character in the hybrid progeny, because in the apomix, what happens? There is no fertilization. Okay, and this apomix, they are produced by the unreduced egg cell. Okay, so there is no fertilization, there is no gamete formation, means there is no recombination and crossing over, there is no meiosis also. Okay, so if these hybrids are made in the apomix, okay, then there will be no segregation of the characters in the hybrid progeny, and the progeny produced will be similar to the parent. Okay, so the hybrid or the desired character will be maintained in the hybrid of one generation after another generation. Okay, if we produce hybrids in the apomix. Clear? Now, then the farmers can keep on using the hybrid seed to raise the new crops after a year, and he does not have to buy the hybrid seed every year. So this is the advantage of producing hybrids in the apomix, because there is no fertilization, no meiosis, no crossing over is taking place. Okay, so the desired character will remain in the hybrid from one generation to the next generation, because there is no segregation of gametes happening here, no segregation of the characteristic happening here. Now, because of the importance of apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of the apomixis and to transfer apomictic genes into the hybrid varieties. Okay, clear? This active research is going on around the world to understand the genetics of apomixis and how to transfer the apomictic genes into the hybrid varieties. Okay, this is clear. So this is the end of this session. I hope you like this session. Thank you very much for listening.